Yeah. So the first category where you have the responders and the monitoring that is built in the responders, you don't need to do anything. You will log into your Brightspace and you start the exam and that's it. For the other category of people who don't want to use the monitoring that is built in with responders, then you have to log into Zoom, the Zoom link that we are using since the beginning of the class. Once you get there, the TAs are gonna look at your surrounding and look at your ID, and then you will get to the exam. And during the exam, for those who don't use the monitoring, they will have what's called Zoom all the time on their phone running. What modules will be included in the midterm? Everything till the midterm. Is the monitoring, is it monitoring our screen or us? It's gonna be monitoring you guys, because the responders monitoring, I don't know if you have used it before. I didn't use it myself for exams, but if you have, if you haven't, the responders, it will look if there is anything suspicious, like you are looking somewhere, you are bringing something in. Yeah. So if something is suspicious about it, then the responders monitoring is gonna give some sort of signal, and we can check it later if something, if the student was not doing something wrong. Now, is the monitoring our screen or you? Gary, is it it's monitoring you, not the screen, because the responders is gonna block your screen. For the Zoom option, we don't need responders. No, Rita, you need the responders locked down. So on your computer, when you log into Brightspace with the responders, there will be no other option on your computer except the window where you have the Brightspace exam. Do you have a link for us to download responders, or it will be on Brightspace for us access before the exam? That's a very good question. In my Brightspace, I see that it's already in there, but it's a very good question, Julia. Yeah, so somebody answered you. You can directly download it. Okay. Should we let you know what option we choose prior to the midterm? Well, I have I have the same set of questions, and I will have both tests for you on the Brightspace. Now, if you go to option 1, it automatically takes you to the responders and the monitoring. If you are not doing the responders and the monitoring, then there's no option for you to get into the exam before you go to Zoom. So you choose by the, like, I really want you to make sure that you know which option you want, because if you use the monitoring, you have to have the camera running on your computer and the responders is gonna take care of you. If not, then you cannot access unless you get first to Zoom and then come back with the TAs, and then they give you the password for the exam. Is this your first exam online using responders? I'm sure last year people did it, and okay, so yeah. So if you haven't done it before, yeah. It's surprising because all the online exams in the syllabus that I read, they should have responders locked down. Anyways, never, Maggie, you are saying never what? I've used responders, but not the monitor version. Yes, because the university gives you the option to be monitored or not. Oh, you have never used before. So that's good. Anyway, you go, guys, go to YouTube and read how responders work as everybody. It's super easy. It's just a click and the responders lockdown is just an option that locks other windows. There's nothing really crazy about it. But you have to have the program there. Okay. Now, let's quickly go to the first thing I want to discuss with you today, which is just a couple of questions as usual. So go to the module number 9, and under that you have the WOOC lab that we would like to display. There are 2 questions there we would like you to answer. So I go share. Okay. Okay. So let's go to the first question. Are you able to see my screen at least? Okay. Awesome. So if you need to overexpress a specific gene, which sequences would you clone out of that gene? Would you clone the introns only? Would you clone exons and introns? Exons only. Exons with the 3' untranslated regions, and exons with 5' untranslated regions. Seventeen people only answered out of 45. You should be ready to answer this within 30 seconds. Okay. So 3% think introns, 8 people consider exons and introns, 45% consider only exons, 13% exons, and 20% exons with the 5'. So the correct answer is gonna be the exons only. And I intentionally give you this question because I went to some of your submissions that you made for assignment number 2, and I could tell that you guys have some confusion about what is a gene per se and what is a transcript. And I will come to a few explanations later on. But the bottom line, if you want to overexpress a gene, you don't want to overexpress the introns because they are not needed. In any case, our body, when a gene is transcribed, the messenger RNA doesn't have any chance except copying that DNA. But after DNA is transcribed to messenger RNA, then the introns are spliced out. So you don't need the exon, the introns. You need only the exons. Now, when you are overexpressing a gene, most of the time we add some untranslated regions, which are the poly A tails in the plasmid itself. If you remember, I showed you on the plasmid, we have the poly A tail. So that's why you don't need the 3' untranslated regions. And you don't want any junk from the 5' end because we just need the start ATG and downstream. So be careful that these processes, that whenever we do the overexpression, we just use the exons. Okay. What is a splice variant? So let's say you have a gene like periostin, and then we have multiple splice variants. It's a mutation in that gene. It's a form of RNA generated by alternative splicing, resulting from a different combination of exons. It's a type of intron retained in the messenger RNA, or it's a duplication of a different chromosome. For those who are still waiting, I don't want you to put your answer just when you see the majority of the class is choosing something. So every time I click, I see many people are trying to click more and more there. You have to evaluate yourself in a way that reflects your knowledge and how much you remember. So it's good. So that's RNA splicing. It means, or a splice variant, it means that it's a combination of exons or parts of exons. Okay. It's not a mutation, and it's not about retained messenger RNA, intron in the messenger RNA. And obviously, number 4 is totally away from the question. Okay, so that's the correct answer. Very good. Majority of you have got it right. Now, these two questions, I meant to ask you those to make sure that you understood what I want in the assignment. And let me go quickly and share my PowerPoint. So, okay. So for today's lecture, we will close up all the information related to DNA technology, recombinant DNA technology, how we use it, and how we deliver DNA into mammalian cells. And we will discuss a little bit about the modifications between the genomic libraries and the cDNA libraries. But before going to that, let me look onto the assignment number 2. So in this assignment, I would like to make sure that you guys are able to go find a gene, get the sequences associated to that gene, and make sure you are getting the right gene. So there were a few submissions that were already done. I went through it quickly, and I tried to track to see what are the major mistakes you're doing. So I just want to let you know that when I ask the question about finding the IGF gene, the first part of the questions are are looking for the genomic sequences. So it is asking you about the gene synonym. Many of you got that right. The number of splice variant, many of you got it right too. Location of the chromosome, these were easy to do, and the direction of the transcript. Now the problem was when I said accession, many of you have wrote the accession for the transcript, which is the messenger RNA or the major transcript. So be careful. The accession number here is related to the gene, not to the messenger transcript, the major transcript. And the current genomic reference sequence, also this is related to the gene. And the genomic sequence information, and I specifically said number of base pairs. And if the number of base pairs is 20,533, it's wrong to say it's a 20 kb. It has to be the exact number because one base pair can make a very big difference for gene transcription. Okay, then question number 8 says identify the main transcript and give the ID for the messenger RNA and how many base pairs, and then how many, the ID for the protein, and then the amino acids. Now, number of introns, number of exons, number of base pairs for the coding sequence. Okay, and here I'm saying design primers to amplify the transcript IGF 202. So if you will amplify the transcript, you are not gonna amplify the exons and the introns. You have to amplify only the exons. So many of you made this mistake. I didn't go through all of you on the submission, but be careful that this question is related to the WOOC lab I just made for you. If you want to overexpress a gene, you take the coding sequence of the gene. You don't take everything of that transcript. And then you need to design the primers. We went through that. But you have to get the right sequence first. So if you make the mistake by choosing the wrong sequence here, like some of you did that, you need to find the coding sequence for this guy, the 202. And after you find that, you design the primers. And finally, you tell me which restriction enzymes you're going to use. And it's not a choice. It's not like, oh, EcoR1 is a famous enzyme and everybody uses it. No, this is not the answer. The answer is to go and look at the lecture, what I did exactly after finding the sequence. You need to know why these restriction enzymes can be used and why not. You have to write, for example, DPN1, I can use it because, for example, it doesn't exist. Or I can't use, I need to know these things. And I want to know that you did the exercise correctly. Okay, so there were already many questions. Direction of the transcript is based on the type of the strand. Greta, so is this the direction of the transcription? Yes, so the direction of the transcription, this is another question, you guys really have to be careful too. Some of you said it's a 3' to 5' end. This is not correct. It's called the reverse strand. It doesn't, it's no way that there will be any transcription from 3' to 5' end. Belkis, you're asking me to repeat what I said about the introns. So the number, I mean, what I said is that when you want to overexpress a gene, you don't overexpress exons and introns. You overexpress only exons. Okay, so go back to the lecture and make sure you look at how I did the things. And this lecture is also recorded. So after the assignment, don't come back to me and say I didn't give you the instructions like you did in the first one. The instructions are super clear now. And on top of this, Greta, you are saying, will the number of base pairs be the same for the messenger RNA of the main transcript? No, definitely not. because the gene is much bigger than the messenger RNA. Many times you see, like, I'll go right now. So let me minimize this, and I will go right now to the NCBI. Okay, so let's forget about this guy here, and let's go to periostin. So we said, for example, you are trying to look at periostin gene. So if you try to get the information about exons, introns, splice variants, it's much easier and more friendly to look at on the ensemble. So I already searched under the human ensemble. So as I said before, the capital letter of the whole name of the gene means it's a human. You already get 4 of your questions answered right away once you click on periostin. It tells you what's the name of the gene, it tells you the synonyms, it tells you where is it on the reverse strand. So if I see some one of you guys saying 3' to 5' end in the answer, don't come to me and I will never consider that correct, because this is scientifically not acceptable. Okay, it's called reverse strand. Then it tells you how many transcripts you have. And if you look, the number of transcripts that is written here, it's already there. These are 10. Now, many of you send me emails, oh, I couldn't find where is the main. I mean, if you click and find main, you already find it there and there. So then the main this means this is the transcript or the splice variant that is mostly studied, and we know a lot of information about it. Okay. Now you already get, every time you click on the gene, you will have another uh, uh, window that will open for this. So now you are speaking about gene. So you see splice variant, transcriptome. You will see a lot of combination of information about the gene, its function, its comparison across organisms, orthologs, references. It's all there. Now, if I click on this guy, which is the transcript number 1, which is the main, it has to open another window there. It's very slow. Okay, it's taken so long, so I'll just go to NCBI quickly to just gain some time. If you are in NCBI and you are looking for the gene of periostin, you have to make sure that it's not everything here. You have to make sure it's a gene. And once you do the search, it will give you the most updated version of periostin. Now, make sure you click on that one. Okay. Now it tells you periostin is a gene. Gene ID is this guy. And then just before, probably when I had the lecture, I was scrolling up and down, but you can minimize those guys. If you need, you can minimize it. And now you want to know more about the genomic region and transcript and the product, which we see most of it is in the ensemble. But you already get a lot of information here. Recall that I put this one here, and you get information about this transcript. But you already know the location on the chromosome and most of the information about the periostin gene. Now you want to get the information from NCBI for the gene, go just click on the gene bank. And it's going to give you a summary. Be careful now that now it's giving you the nucleotides of that gene. But if you read it, it tells you Homo sapien chromosome 13. So it's the location in the chromosome. The reference sequence for that gene in the genome is NC 0004013.11. It tells you where it's located, the locus, which it happens to be the same like the accession. And then it tells you how many base pairs of DNA it is. So that's the genomic DNA. So that's the genomic DNA for the gene periostin, right? Now, if you want to know more about the splice variants, it's really not that friendly on NCBI. But if I go back one step and simply minimize these guys, there are genomic products, there are transcripts and other products. So the transcripts are listed here. Of course, there's too many. It's so difficult to navigate here. That's why I said, better you do the transcripts. That's what I do under Ensemble, because if you want to know about the transcript 203 for periostin, once you click on it, it's going to open this window here and the interface changes there. You get exons, you get coding sequence, you get protein. This information is also written there. You have the how many base pairs it needs, it is, how many amino acid it produces, the coding sequences, and the reference for the messenger RNA. Now, if you click on that one, it takes you to a page that is similar to what I've shown you for the genomic DNA, but now it's saying Homo sapien periostin, transcript variant 1, a messenger RNA. So that's a messenger RNA. And that's a reference sequence for the messenger RNA. Then if you look down, it tells you the accession for the messenger RNA. And remember, I told you the NM means messenger RNA. So if you write the accession number NM for the genomic DNA, that's totally wrong. Go back to your submissions, and if you made this mistake, correct it. And then it tells you the base pairs and all that. And it's saying that this is the main select. Also, if you look in Ensemble, it also tells you it's the main select. And if you scroll down, it tells you how many exons, how many coding exons are there, how many base pairs, how many amino acid residues, and it gives you all the information you need about the reference sequence for the messenger RNA, the reference sequence for the protein, the coding sequence itself. So when you do your primer design, that's where you need to get the coding sequence for that question in the assignment. You either get it from here or you get it from there, or if you are in NCBI, you get it from all the way down here, coding sequence. Okay? I hope this clarifies the situation for you. And I will stop to take any questions before moving on. Okay. So what is the accession number? I think I answered this question. Is there another word for accession? Because I've only seen it in it for messenger RNA. I just showed it to you, Kenan, on the genomic DNA as well. Beta, can you please go back to the recording and watch the steps, because I really need to move on. So Let me see if, well, okay. Let me share this again. Let me share this again. So stop sharing this guy. Okay. Let me share this again. So that NCBI for one last time. For the NCBI, you have to make sure you're looking under the gene for the gene name periostin, and it will pop out the list of genes you have. It will tell you the most studied one. If you click on that one here, the periostin, which is the top, if you click on that one, it takes you to a new page where you still are under gene. It's telling you the information. It's telling you the link between the NCBI and the ensemble. Just minimize these because most of this information you don't need. If you click on the gene bank for this guy, it takes you to the genomic sequence for the DNA. And for those who said they can't see accession for DNA, look at this. This is the accession for the DNA. And after that, when it says which version it is, I mean, all you have to do, guys, is just Google or control find, and then you will find this information. And I'm sorry to tell you, I figured out many of you didn't really go to the lectures. Some of your answers were really so far from what I discussed in the lectures. Anyways, I'm trying to make sure that everything is pretty much clear for you so that you get the best points out of the assignment number 2. Assignment number 2 is big, 15.5 points. Almost every question is 1 point or a little bit less. So make sure you guys work hard on getting this done correctly. Because later on, if you lose one question, you're gonna lose 1 point, and that's not gonna be good. Okay? So stop sharing this guy and share the presentation. Okay. Now, any questions regarding the classes we went through, the lectures, the sessions we discussed up till today? Okay. I don't get anything on the chat, so I'm assuming that everything is good. Joe, you have to go back to the lecture to see how we find the forward and reverse primers, because we discussed this extensively there. Okay, so up till now, and that's the roadmap, how I planned the sessions. So in the first session, we discussed the cell culture. In the 3 sessions after that, we discussed how to extract DNA, detect it, run it on a gel, check the quality of the DNA, if it's degraded or not. Then if I want to amplify a specific DNA, I know how to design a primer for a PCR. I know how the sequencing work. If I don't know the sequence of the DNA from 0, I can do it. I can get information about a certain gene from genome browsers like Ensemble and NCBI, and this is very, very shallow. You could study Ensemble and NCBI for hours and hours. We know how to use restriction enzymes in order to clone this DNA into a plasmid or other type of vectors. And then we can deliver it to bacteria, so we can have a clone, so we can grow the bacteria, get amplification of that DNA and reuse it the way we want. And today, we're gonna close up by delivering that DNA or plasmids or vectors into eukaryotic cells, like mammalian cells, like HeLa cells, 293 cells, cancer cells, whatever. So pretty much we cover a lot of information about the DNA itself. And in the next sessions after the midterm, we will discuss how we manipulate RNA and proteins. Now, in the previous lecture, so let's go here. So in the previous lecture, we discussed the transformation, which is the process by which we introduce DNA or a plasmid into bacteria, either by chemical transformation, having some calcium chloride, then heat shock, or by electroporation, where we introduce a sort of uh, electric shock that causes small pore formation within the bacteria. The DNA gets in, and we can select based on the plasmid being used. If we have resistance for ampicillin, we grow the bacteria in ampicillin media. Only the bacteria that have the ampicillin resistant gene through the plasmid would survive. Now, we discussed about the steps, and we discussed about the generation of recombinant proteins like insulin. So I had a question from some of you about pcDNA3. I don't have it on my slides, unfortunately, today. So if you stay on at the end of the lecture, I can show you and discuss a little bit more about pcDNA3. So we mentioned that the recombinant DNA technology is a wide range of applications that can be used in industrialized applications. Like now we know that we can generate a lot of proteins like insulin and other growth factors, cytokines by bioreactors, the way I described in the last lecture. And most of these are based on information that we know that we need some sort of clinical intervention. Like we can look to identify which genes that cause diseases by working with the recombinant DNA technology. So in other words, if the gene is mutated in a human and we detect that a population of human uh, get a mutation while other people don't get a mutation, and those people who have a mutation has a certain disease, then we can link that mutation to the disease. So we can isolate that gene, look what the mutation does, and all this. So the industrial and the clinical application are strictly based on the basic research application. So first we have to make a theory about what is this mutation causing, for example, or why we have multiple splicing variant of that protein. And then we start discussing, oh, this splice variant can do this splice variant can do that. So we have to clone them, overexpress them, or delete those types of proteins. So the basic research is the first building block to get to any of these clinical or industrial research. Because obviously, when people knew that the insulin deficiency is the major cause of type 1 diabetes, there is a lot of research about insulin itself that identified the gene of insulin, identified which sequence should be amplified in order to produce insulin, and what are the other factors that are needed for the insulin to be functional. So we get this in the basic research. And then we link the basic information we know about insulin and pancreas and the physiology to the clinical implications, where we saw many people, for example, lost their beta cells and they are suffering from type 1 diabetes. And therefore, beta cells are producing insulin, and that's how the linking happened. Then we ended up producing the insulin in an industrial scale, because now we know there's a majority of people who have diabetes, they need insulin, so therefore we use the RNA recombinant DNA recombinant technology for the generation of this. So this is a very, very long way, but the steps are very essential, that the ones that I described for you in terms of the cloning, overexpression, and the transfections. So there are many other applications that we didn't discuss last time, and I'm just giving you a small summary about that. So the recombinant DNA technology can be used in protein production, and insulin was one of the examples, but there are another 170 other proteins that are being overexpressed right now and being used in therapeutics. So there's a complexity of lots of these factors, like for blood clotting, for other enzymes that can be used in many, many diseases. So insulin was one example that is giving you the details. DNA technology is also used in gene therapy. And when we say gene therapy, this means there is some sort of change in the genetic code or the genetic expression of certain gene, that somehow this change in the genetic information could lead to a change in, obviously, in the RNA and the proteins downstream from that gene, transcribed and translated from that gene, and then probably that can cause or treat a disease. So the gene therapy could be divided into two major fields. The first one would be the gene transfer therapy, and in this case, you are introducing a gene into a cell. And now the vectors that you are introducing would be correcting a certain function within a gene. Like, for example, in cystic fibrosis, the copy of the gene CFTR, if it becomes mutated or dysfunctional, it causes the cystic fibrosis. So nowadays, we could overexpress a form of the CFTR gene that could correct the function, and that will replace the defective version of that gene. The novel versions, and this is a really very fast growing field where we do the genome editing. And in this situation, we will introduce some changes to the genome, and that's mostly mediated by the CRISPR-Cas9. The genome editing, we will have one session by the end of this semester. It will be 100% dedicated to the genome editing, because that needs one whole lecture by its own. But the idea is you can modify a gene function either by adding something or removing genetic material from the genome. And nowadays, the CRISPR-Cas9 is the most widely used, and it is very, very novel. And so we use this in animals, obviously, but in human, that is still not the case. So in order to knock out a gene or overexpress a gene in genetically modified animal, this has been done a long time ago. Now, for cystic fibrosis, as an example, so for those who have a mutation in the CFTR gene, which stands for the cystic fibrosis transmembrane conductance regulator, these cells are responsible for the regulation of the synthesis and the thickness and the quality of the mucus on the lining cells in different organs, including the mucus that is generated in the respiratory system and in the digestive system. And whenever there is a mutation in this gene, then there is a change in the thickness of the mucus. So if you look at the airways of a healthy person, compared to cystic fibrosis, the thickness of that mucus, it causes the airways to be smaller. So the diameter of the airways are smaller, and therefore the ability of air exchange is less. There are many versions of the cystic fibrosis. Some of those is that no protein synthesized or not enough protein synthesized, or the protein is produced, but it's not the correct uh, format. In other words, it could be like not functional protein that is produced. And all these could lead to the same outcome physiologically, that the mucus is very thick, and it's not only in the lung, obviously in other organs like in the intestine, and this would cause two troubles. So now this nowadays can be corrected by recombinant DNA technology by introducing the correct form of the CFTR. Now, if I stop a second here and we can go and watch this guy. Okay. Okay.
Promising therapies are being developed to treat the genetic cause of cystic fibrosis. When delivered through the airway directly to cells, these therapeutics have the potential to restore CFTR function in the lungs. Highly specialized cells that line the respiratory tract help protect airways from invading microbes, infection, and injury. At the innermost layer, basal cells are constantly replacing damaged and aged cells. Secretory cells release mucus into the airway to trap foreign particles, while ciliated cells move the mucus up and out of the lungs. Ionocytes are thought to express high levels of CFTR protein. The instructions to make CFTR protein are encoded in the DNA of each cell. The CFTR protein helps control the movement of salt and fluids into and out of the cell to keep mucus at a healthy level. With cystic fibrosis, DNA mutations create either abnormal or no CFTR protein in the cells, causing a buildup of thick, sticky mucus. By providing the correct instructions, it is possible to reprogram cells to make normal CFTR protein. Correcting CFTR expression in even a portion of the cells could be enough to restore sufficient function. Two potential treatments are RNA therapy and gene therapy. Both approaches can deliver a correct copy of the genetic instructions to cells in the lungs. With the correct messenger RNA or DNA sequence, cells would be able to produce normal CFTR protein that moves to the cell surface. There, it can adjust the amount of salt and fluids flowing through the cell membrane. Over time, the correct copy of the genetic instructions is degraded or lost as the cells naturally die. Regular repeated treatments will be needed to ensure enough CFTR protein to hydrate mucus. Gene editing is a potential therapy that would permanently repair the CFTR gene mutation in cellular DNA. The goal is to specifically target basal cells because they have the ability to divide and differentiate into other cell types to replenish lost cells. As cells die off, the basal cells with permanently repaired DNA generate new cells that contain the correct CFTR gene sequence. If enough basal cells are successfully edited, cells with normal CFTR will constantly be supplied to the airway surface. Repeat treatments would be rarely required, if ever.
Okay. So at least this gives you an information about how that works. Okay. I see there were some questions. Is it true, as you explained Tuesday, it doesn't matter where we base our primers off as long as the conditions are correct? Because Miranda today you mentioned the importance of exons. Miranda, so the question was clear. If you are trying to amplify a piece of DNA, if it's a 3 kb or let's say 3000 base pairs, it includes 5 exons, and you need these 5 exons. So it's definitely you need to have the start at the end where your primers are. Now, based on this information, that's how I will look where you design your primers, because I asked you to specifically amplify the transcript 202 in that assignment. Therefore, you have to go and find where are the coding sequence for the 202, and your primers should flank all the sequence of that coding sequence. Okay? So now let's continue. So other applications of DNA recombinant technology is the vaccine development. And as I described before, the functional genomics to understand how the genes work. So in the vaccine development, you can tell that, again, every time we go one step ahead. Imagine we have a virus that is infecting the human. Take this example, let's say COVID-19. We get that virus, we get all the sequences from that virus, and then with bioinformatics, we would say this part of the virus probably can help us to design a vaccine toward the virus. It will recognize only that virus. So imagine we found 3 different hits that say probably this part, probably that part, probably these 2 parts together. So we get the genes for those guys and we clone them. And then we overexpress those proteins. And after that, we inject those into mice or animals. And once the mice react to that protein, we would be able to take the serum and isolate the antibodies or the vaccine. Now, I'm not trying to ask you questions about the vaccine itself. What I'm trying to tell you, if you have a virus that you don't know anything about, like COVID-19, and we would like to get a vaccine, you have to get the DNA and you have to get comparison between the viral DNA and other viruses so that you get very specific genes to this virus, and then you have to clone these genes. So that's what we exactly studied so far. Now, those steps, I'm not going to really ask about that. The other thing is, when we discuss the functional genomics, this means we can clone into vectors those genes, and we can either overexpress the genes, means make many, many copies of the same gene, or we can downregulate the gene, means we can make lower copies of that gene in one way or another. And again, gene silencing is one way where we do downregulation. So by using RNA interference or CRISPR-Cas. And when we do the downregulation, there are 2 outcomes. We can either knock down the gene, means we can partially delete the protein. In other words, if you have 100% capacity of a gene, if you knock it down, it will still be partially functional. Some of the proteins will be still there, or we can knock out the gene, and in this condition, we will have a complete deletion. So all that is just applications of the recombinant DNA technology. Now, how do we transfer a piece of DNA that we clone into a plasmid into our cells? As described before, in bacteria, we had either the chemical transformation or the electroporation. Now, it's more difficult to deliver cloning vectors into eukaryotic cells compared to bacteria because the eukaryotic cells are more complex. First, because they are in a growth media, so bacteria are way different. The growth media is different. The way we can freeze the cell culture for the eukaryotic cells is way complicated compared to bacteria. And obviously the complexity of the cell in general, the system in general. So electroporation can be used in the same manner that we used for the E. coli transformation or bacterial transformation. We can use electroporation, where simply the cells are mounted with the DNA and then an electric shock will be added. Of course, this will make polarity, and since DNA is negative compared to a positive intracellular part of the cell, the electroporation, as I said before, it will generate mainly some pores in the cytoplasm. Once the shock take place, these pores allow the negatively charged DNA to be integrated into the cell. And after the heat shock, when the recovery period takes place, the membrane will be recovered. Of course, too many cells will die when you do this, but some of cells will survive. And these are the cuvettes that we use in the lab. You see that they have two sides here, one for negative and one for positive, and that's how the electroporation takes place by creating a current, a negative to positive. And all you need to do is have the cells and the DNA in that cuvette before giving the electric shock. It's like 15, like, I don't want to say 15, it's like 1 second, like this. This has been used previously to generate transgenic animals by injecting DNA directly to the fertilized oocytes. And the major way, as I said, is you create that electric field will cause the generation of certain pores within the membrane, and then that will lead to the DNA entry. And all you need to do after is just to recover the cells from this shock. And again, a lot of the cells will die. So this is one way to deliver DNA into mammalian cells. The other way is the chemical transfection. And in the chemical transfection, these are mainly depending on the negative charge of the DNA. So if we look here, the first one is the calcium phosphate. So it's a specific buffer that contains calcium phosphate. And since the calcium is positively charged and the DNA is negatively charged, then you will have this sort of neutral molecule that forms here and somehow get integrated into the membrane and passes through the like an endosome. Similarly, the cationic polymers, again, they are positively charged, will wrap around the DNA, similar to what we see with the calcium phosphate. But the most used ones and the most efficient are the lipofections. So this is a form of the chemical transfection where the nucleic acids form with the lipofection reagent a liposome where the DNA is already within the liposome. So all you need to do is to get these lipofections, mix it with the DNA, you get these type of liposomes. And because of the nature of the plasma membrane that is similar to the liposome, then it will be much easier to fuse with the DNA, with the membrane, and pass into the cell. And this is an example where different types of cells were transfected with the plasmid to overexpress the green fluorescent protein by using the lipofectamine, which is one of the lipofaction reagents. And the third is the viruses. And this is one of the most efficient ways to introduce genes to the cells. Broadly, the viruses, I'm just giving you a summary here. Again, this is really, really shallow. We can divide them into two major parts, the retroviruses, and these are viruses that use the single-stranded RNA as a genomic material, compared to adenoviruses, which use the double-stranded DNA as a genetic material. And therefore, for retroviruses to become functional, the first step would be is to reverse transcribe the RNA into DNA. So they need to have a reverse transcriptase enzyme that is capable to generate the complementary DNA from the viral RNA genome. An example of those is lentiviruses, and the most widely known is the HIV, the human immunodeficiency virus. When we say lentiviruses, the word lenti means it has a long time of incubation. So the virus will be integrated into the genome and it will have a long, long time integrated in our genome. The most commonly known adenoviruses are those that affect the respiratory system that can cause cold and flu. So for the sake of this class, we will discuss only lentiviruses and how they are generated. I'm not going to discuss adenoviruses, but I have references for you if you are interested too. So let's look at the life cycle of a virus. The virus is not alive as much as it's outside of a host, whether this is a bacteria or a mammalian cell. So it's just a flying particle. We call them viral particles. These viral particles has on their envelope certain proteins that interact with cell receptors. And that's how the viruses select their host. Like that's why COVID-19 was specific for the lung or the respiratory system in general. Then once their virus binds to the cell on a receptor on a cell, it fuses and gets integrated into the cell. And the moment it becomes internalized, the reverse transcription will start. So if the genome is RNA, it will be reverse transcribed into double-stranded DNA. After that, that DNA that is generated is gonna translocate into the nucleus, where it gets integrated into our genome. There are a lot of studies and complications how the virus do that. I'm not gonna go into that discussion. And where it gets inserted and what it does after it gets inserted. There's a lot of complications. But then after the insertion take place, the transcription and the translation of the viral proteins start to happen. And now this particle has a part of our genome that is producing everything that the retrovirus need to be regenerated again. The proteins, the enzymes, everything that the virus needs are going to be generated from that genome. And now, after generating all these proteins, the virus starts to reassemble itself again. And the final steps of the reassembly take place at the surface of the cell. Once there are many, many numbers of these viruses, the budding start to take place. And most probably that's the time when the cell will die. Because what we do in the culture, in the lab, the cells that produce the eventually are so sick to the level that they are dying. And that's exactly what happened in physiology, in regular physiology in diseases. When someone gets infected with a cell, a lot of these cells will die by the time the virus is being generated and the new viral particles are going to be released. And obviously, these particles will go and repeat the process again and again. Now, there's a misrepresentation that you have only one virus here and 3 viruses there. It could be like 1 virus get here and 3 million get out there. Okay, just don't get the representations wrong. There are more information for you here if you want to know about the adenoviruses. Now, take into consideration that we know these steps. We trick the viruses and we use them as a mean so that we can take a retrovirus, we can give it the gene that we are interested in, whatever that gene is, we want to study, overexpress, downregulate, or whatever. And then we infect the cells and we can study the function of that gene. So in order to do that, we use a complicated set of cloning to generate a virus. So there are several types of the viruses, as I said. The lentiviruses is the one we are using here, although there are retroviruses other than lentiviruses and adenoviruses that we are not going to describe. So the viral particles are obtained from the culture by virus packaging cells. So again, if we go back, if this cell is able to be infected by the virus, in other words, it can make a packaging source for us to generate many of these viruses. And that's exactly what we do in the lab. And in order to do that, and for safety reasons, we put the major genes for the viral production in 2 different plasmids. And we put one plasmid where we can insert the gene of interest. So we need the cells and the 3 plasmids. So first, we have a transfer plasmid that contains the gene of interest that you want to study. We can clone whatever gene into that plasmid number 1. Then we have the packaging plasmid and the envelope plasmid. These are the 2 plasmids that gives the genes needed for the virus in order to generate the packaging, encapsulation, and the genome of the virus. Like the most known are the GAG, POL, REV, TAT, and these are the genes here. Each one of those has a specific function. And then we need the VSVG. Remember this gene that is important for the binding of the virus to the surface of the cell. Once we put all these 3 cells together into the same cell, then that cell will act as if a whole virus came in and all the 3 parts of the genome are integrated. And therefore, we are taking as if we are at this step. When we transfect all the 3 plasmids together, we will get viral particles start to be produced. And the packaging cells, which are in this case 293 T cells, will produce infectious particles, which are the viruses, whose genome is now, we have tricked the virus. In principle, instead of carrying its own genes, it's carrying our gene of interest that encodes for a sequence from the transfer plasmid, which is the target gene that can be used. And now, if I want to transfect, if I want to study that gene, all I need to do is to take that virus and imagine I want to check what it does on HeLa cells. I will take that virus that I produced and I will infect HeLa cells. Or I want to infect cells from cancer. I take that virus and I infect it and I put it on cancer cell and I will see what will happen to that. Now, one thing I have to do, obviously, if the way we generated those viruses is to generate this gene particularly. So we have engineered the viruses in a way that they don't produce more viruses in the lab. This happens in the natural condition here. But what we do in the lab, we blocked this part there. So when we infect HeLa cells after producing a virus here, we will not have a regeneration of the virus. We will have only the overexpression of this target gene. Okay? I think we are good enough to stop and take some questions. Is the DNA marked to see which cell contains the DNA? Gary, can you please repeat your question? I didn't get it.
Yeah, I was just referring to in electroporation, since not all cells take in the DNA and die. I was just wondering if you mark the DNA to see which cells take in the
So
DNA.
there are, if the plasmid has a selection marker, you can add the antibiotic or the selection marker. Like we remember the G418. If you add the G418 later on, the cells that contain the plasmid that has the G418 selection will survive, and the cells exactly like the bacteria. The bacteria that had ampicillin survived, ampicillin resistance survived, and the one that didn't have the plasmid with the ampicillin resistant, they die. The same way on the mammalian cell, you can add the G418 on the culture media. The cells that were transfected with the pcDNA plasmid, let's say, they will survive, and the one that didn't get any plasmid, they will die. Now, Joe, you are asking whether would you like us to know the lentiviral cycle for the midterm? Um, yes. Yes, I would like you to know that part. Any more questions? And let me go back to Miranda's question, because this concerns everybody regarding the assignment. Did I understand your question correct, Miranda? You said, is it true, as you explained Tuesday, that it doesn't matter where we base our primer off as long as the conditions are correct? No. The answer is no. Again, in this particular question, you have transcript 202 to be overexpressed or amplified. So in order to amplify the whole part or the whole coding sequence of transcript 202, you first need to get the coding sequence. And your primers should flank the beginning and the end of that gene. So Julia, you are asking everything up to here. We ended the lecture today, will possibly be on the midterm. Yes, everything till today will be a part of the midterm. On Tuesday, there will be no lecture. It will be just the quiz to set up for the Thursday midterm, and you are free to ask a few questions if you like. I'm not going to stay the whole lecture because I have a meeting in the department later on. So Maggie, you are asking if this was already answered. I may have is, but is the midterm all MC, multiple choice? The midterm will be multiple choice and true or false. I will send you a detailed email about this. Joe, you're asking how many questions, how much time? You might get up to uh, 40 questions up. I didn't really decide, but more or less you have 30 points, so it could be between 40 and 50 questions. The midterm will be, are we using responders for the quiz? Yes, Julia, we will use the responders and the Zoom. So for those people who has responders monitoring, meaning they will accept that, uh, it will be only responders And for the ones who have, yes, and for the ones who do not want to have monitoring automatically, you will get first to Zoom and then you will go to the Brightspace. Yeah. Any more questions? Is everything clear for the assignment? You guys know what you have to do in order to get the full point for the assignment. And for those who submitted, please go back based on this, based on the lecture today and based on the information, go back and check your answers if this was correct. Now, I'm not saying that one of you particularly made it wrong. I went quickly and I looked for many of you and I picked some of the mistakes. Raul, you're asking if the assignment, if our reverse primer has herpin, what do we do? Explain how you can fix it or fix it. For question 8, I'm kind of confused of the exact answer you are looking for. We are looking on the messenger RNA sequence and the protein sequence. Joe, if you let me go back to the question. Question number 8. Identify main transcript, main select transcript. And then it's very clear the question, Joe. The ref sequence match for the messenger RNA and how many base pairs? The ref sequence for the protein and how many amino acids? Is Tuesday quiz for practice? No, Pida, the Tuesday is going to be only 2 questions and you can earn 1 point for that. So it will be graded. Okay, guys, seems that all set. Gary, please go back to the review that I made because I answered this already 3 or 4 times. Gary, the current genomic sequence is referred to the version. Okay, guys, no more questions. Anybody has any other question? Otherwise, thank you very much. Questions, comments?
Hi, Professor. I have a question about assignment 2.
Mhm.
So I know you showed us through the Oligo website, I think, how to check for self-complementarity for our primers.
Mhm.
But how do we check if both are primers, if there's a chance for cross-dimerization?
For both,
Yeah.
you can double check with the primer website. If you
Like
look at the end of the, if you look at the end of the assignment, I put you a lot of resources that you can use. So you can pick the 2 primers and you can check if they are self-aligned there.
Okay. Okay, thank you.
Sure. I think it's called primer 3 plus or something like this. I have I have who raised his hand here?
Hi, Professor. I was just wondering if cDNA library generation is going to be covered on the exam?
Um, Probably if I cover this next Tuesday. If not, I'm not gonna ask anything that I didn't cover in the class.
Thank you.
Thank you. Jacob, you have a question? Diana, Muna, Nadia, I can still see you guys here. No, you're good. Everybody's good.
Sorry, I just had another question. About the introns on the website, like where it showed, it would like go through a range, like it would be like 1 to 2 or 2 to 3 introns. Do we just count like the total of them?
Yes, how many introns and how many exons. So
Okay. Okay, thank you.
yeah,